Hi, I'm Peter Cook. Welcome to the pan-genomic model, part 3. In part 2, I suggested the classical mechanism drives DNA looping and clustering. So we have clusters of transcription factors and polymerases that emerge spontaneously without energy input. Additionally, small world networks also do so. Social networks are small world. I might know somebody in New York who knows somebody else in Australia who knows another person in China who in turn knows me. This network might overlap with another that all share an interest in a particular football club. I'm now going to describe some small world networks detected using simulations through a collaboration. Now, biologists are usually skeptical about simulations unless they're based on the simplest possible set of assumptions, and then they can define the limits imposed by physical laws. So as a biologist, I find them very useful to start thinking about complexity, especially when these simulations replicate structures found in vivo. We'll begin with red proteins that might be transcription factors or polymerases binding reversibly to pink DNA beads. All of the other beads in the string are not going to bind these red factors. So a red factor might bind, and then I want you to watch what happens when 3 dissociates. And to have a memory of the original structure, I'll duplicate that structure and we'll watch what happens as 3 joins the cluster on the right. Now I'll build a network. If we focus on this cluster, then 4 and 5 might be being transcribed. Then we would say 4 and 5 are on together, and we represent that with a black edge. These 3 are interconnected by another set of black edges. And similarly, here, 1 and 2 are going to be connected, as are 3, 4, and 5. Now, these 2 structures might represent the same cell at different times, and we'd like to take an average of the 2. This is because they could represent different cells in a population, and then we take the average just by overlaying one over the other. And of course, RNA-seq and Hi-C generally take population averages. This network is a small world one, and one way of looking at the small world nature is to say that 1 and 5 are far apart on the genetic map, and yet they're connected by only 2 black edges. And it's well known that gene expression networks are small world ones. You will recall that I said before that GWAS can be viewed as pointing to complex regulation when thousands of loci around the genome that do not usually encode proteins or factors control the activity of your favorite gene. But in contrast, Yamanaka's experiment seemed to say that just 4 factors could switch cell fate, pointing to a much simpler explanation. I'm going to illustrate these experiments by reference to one particular human chromosome and a small portion of it. And this is because the network is already very complicated. The black edges represent positive correlation, where the 2 pink beads are going to be on together. But of course, in some situations, one is on and the other is off. In other words, there's a negative correlation, and they're shown as the gray edges. So it's definitely a small world network where Many lines connect each bead to other beads, and it's consistent with the GWAS results. But thus far, in all the simulations that I've described, the factor concentration is approximating that found in vivo, where the number of factors is smaller than the number of binding pink beads. But recall that in Yamanaka's experiment, he overexpressed factors. So we'll do that in the computer and raise the concentration of factors only 2.4-fold. And now you can see that the network has become much simpler. Initially, we have a very, very complex network consistent with the GWAS results, but overexpression simplifies the network so that now the 4 Yamanaka factors can play their decisive roles. In other words, we've reconciled the results. There are surprising long-range effects that are seen in these simulations, which I'll illustrate. If you look at the cluster on the right, both pink beads are being transcribed. Now let's imagine that we make a point mutation that knocks out the binding of one factor to a bead so that this transcription unit ceases to be transcribed. And so this knockout destroys this cluster, and the remarkable finding is that other transcription units, both near and far away, change their activities. So we now repeat the experiment by knocking out in turn each pink bead in a long string, and we find that on average the contacts and the transcription of about half the pink beads is affected by a single knockout. And this, again, is in accord with GWAS results. Now we're going to make things a little more complicated. We're going to have 2 sets of factors. We're going to look at the overlap between the different networks. We've gone on to carry out simulations with 5 different kinds of transcription factors, and then we obtain 5 different kinds of color in the clusters with very little overlap between the different colors. Now, small world networks have been detected using experiments. It's a universal finding that co-regulated genes are found close together in 3D space only when active. And the earliest example came from polymerase I genes. In man, for example, ribosomal DNA genes are active in nucleoli. Some chromosomes may not associate with the nucleoli, and then those ribosomal genes are always inactive. We then showed that polymerase II sites in the nucleoplasm were quite distinct from polymerase III sites, and we call these distinct sites factories. But a factory just equals a cluster. And we began to show that different transcription factors were concentrated in different kinds of clusters. For example, genes that tend to be spliced tend to congregate in different places from those that are not spliced. Similarly, genes that are regulated by the transcription factor ER-alpha, genes that are involved in globin production, and inflammatory response genes, all are concentrated in local clusters when they are active. And those are just some of the early examples. So I'm going to reprise this in a slightly different way. In this 3D view, we imagine that the pink clusters might be on together, and there might be many of them in the nucleoplasm, and so there's a small world network of pink promoters. There's another group of orange ones, and so on. And we'll look at this now in another way. You will have seen many plots like this, where each dot represents the transcripts that are found in a different cell. In this case, they're taken from mouse blood. The group of transcripts in cells can be grouped together so that neutrophils tend to contain transcripts of the same type that are different from those found in macrophages. If you drill down, you find that each type of cell has its own distinctive pattern of transcription factors. However, there is a continuum between the different cell types. It's this that we need to explain. And I'm going to call this continuum a blending, and you'll see why in a moment. So I've introduced you to the idea that small world networks are very, very complex. In our simulations, we've just got 2 colors, but in reality, there are about 1,600 transcription factors in the human genome. These factors can act as activators and repressors, or both. And it turns out that most transcription factors, which had previously been called activators or repressors, are actually both. And whether they're one or the other depends upon their precise position relative to the transcription start site. And there are many binding sites. And I'll illustrate this by reference to 2 widely known genes. And I just went to 1 kb around the promoter and asked how many different transcription factor binding sites were there present. In both genes, only a tiny fraction are occupied at any one time. So I think you can immediately see that there are an incredible number of combinatorial possibilities, which I'll illustrate just using a simplified gene with just 4 sites. One might be occupied, another, or 2, or 3, or 4, in different combinations. Moreover, most predicted binding sites play no functional role. Now, thus far I've been talking about DNA binding sites because we all used to assume that factors bound to DNA. No, it turns out that most factors also bind to the nascent RNA through a conserved motif. This is a long-winded way of saying that decoding how factors work is a major challenge. It's such a major challenge that it even has a theorem named after it. Things are just too complicated, so perhaps we should give up. Of course, I'm not going to give you a solution to this problem, but just 2 approaches. The first is going to be bottom-up. We perform simulations, and these are the ones that I've shown you. We had 2 colors that bound specifically to their cognate beads, and they gave 2 kinds of clusters. All the reds were in one, and all the greens were in a different one. Now we're going to repeat the experiment. We're going to say that factors not only bind specifically to their cognate sites, but also non-specifically to all the other beads in the string. And this is a perfectly sensible assumption because this is exactly what factors do. And then we find that some clusters are now mixed. And there's a huge range and variety that depends upon the precise numbers and relative affinities. So I'm going to describe this as a blending between the 2 colors. If we now turn to the human genome, it is easy to imagine that there are many, many thousands and millions of blends that are possible. So what we have done with these bottom-up simulations is to say that the rich complexity that is seen emerges once again spontaneously from simple assumptions. There's also a top-down approach that most people are using. You just ask your favorite AI or machine learning program and apply it to data from as many sources as you possibly can and seek the answer. Of course, you don't really know what's happening. I think everybody will agree that progress is being made, but it's very slow, and I recommend that you read this review. So clearly, decoding how factors work is a major challenge. Fuzzy logic that is embedded in the DNA sequence of the binding motif, the factor concentration and affinity, and 1D and 3D position, both near and far away, on the same or different chromosomes. And this fuzzy logic is going to integrate all these different inputs. What I've done today is to describe some small world networks where co-regulated genes cluster when active to form networks. And I've described how these networks are incredibly complex. In the next section, I'll describe some properties of these clusters that we called transcription factories. Thank you for your interest.